Evolution and statistics. In this activity, we will complete a scenario together to help us better understand the Hardy-Weinberg principle and its application in predicting characteristics of populations. Activities like this help us grasp the concept of genetic equilibrium and how it can be used to detect evolutionary changes in populations. And understanding these principles is crucial as they provide a foundation for studying genetics, as well as evolution, population dynamics, which are all applicable to various fields such as medicine, agriculture, conservation biology, just to name a few. So by learning to analyze and interpret genetic data, we can develop critical thinking and problem-solving skills that are essential for everyday life, in addition to scientific research. So you are already familiar with the criteria for equilibrium based on Hardy-Weinberg principle, but another aspect of the Hardy-Weinberg principle is a set of equations that allow us to predict characteristics of populations at equilibrium. When a population of organisms is at Hardy-Weinberg equilibrium, it is possible to make calculations about allele frequencies and genotypic frequencies based on the following equations. The first one being p squared plus 2pq plus q squared equals 1. Let's break down what this equation really means. When we look at p squared, that represents the frequency of homozygous dominant organisms in the population. Usually, we'll break down exceptions in just a second, but generally speaking, p squared refers to the frequency of homozygous dominant organisms in a population. All right, so thinking through it, right, if the genotype is big A, well, big A times big A is the same as A squared. So that's how we get P squared for representing our homozygous dominant organisms. Then we have 2pq, which represents the frequency of our heterozygous organisms in the population. And again, thinking through it, if the genotype is heterozygous, individuals could be either big A little a or little a big A, right? And big A little a plus little a big A, well, that's equal to big A times little a plus little a times big A, or 2 big A little a, or 2pq, okay? And then lastly, we have q squared, which represents the frequency of homozygous recessive organisms in our population. So thinking through that last one, right, similarly to our p squared, our q squared, if we were to have the genotype little a, well, little a times little a is the same as a squared, okay? All right, so that's our first equation. Our second Hardy-Weinberg equilibrium equation is p plus q equals 1. And this makes sense, right? If we're looking at frequencies, all of these represent frequencies, so the sum should always equal 1, or 100% if you were to convert it to percentages, right? And so if we have p plus q equals 1, what does this really mean? Well, breaking it down from our previous equation, right? P by itself represents the frequency of dominant alleles in the population, while Q represents the frequency of recessive alleles in this population. All right, but note, and here's the exception I was referencing earlier, if you are not dealing with simple dominance inherence, P and Q can be arbitrarily assigned to allele frequencies. Now, both of these equations add up to 1 since we are using frequencies. If we add up the frequencies of the homozygous dominant organisms, the heterozygous organisms, and the homozygous recessive organisms, that would be 100% of the organisms in the population, right? So a frequency of 1. A frequency of 1, remember, equals 100%. If we add up the frequency of the dominant alleles and the recessive alleles, that would be 100% of the alleles in the population. So again, a frequency of 1 equals 100%. The Hardy-Weinberg equation allows us to predict the genotype and allele frequencies we would expect to see in a population that is not undergoing evolution. Remember, a population at equilibrium is not evolving. Equilibrium opposite to evolving. This is useful, though, because if a population differs from the expected frequencies of these Hardy-Weinberg equilibrium equations, we know that the population is undergoing evolution. So for this activity, we will be using a new statistical test to compare the expected number of individuals with each genotype in a population, so the frequency we would expect to see based on Hardy-Weinberg equations if the population was not evolving, to the observed number of individuals with each genotype, or the frequencies we actually observe in the population, to determine whether this population of organisms is evolving. And just as a little side note, this scenario is actually inspired by a real natural phenomenon covered in a paper that we have linked to in Canvas. And this is an example of primary research, so a really fun paper and reputable paper to check out. The scientific data and inheritance patterns of snail genetics have been simplified, though, for this activity. So let's go over the scenario. What are you studying? The scenario. You are a researcher studying the impact of urban heat islands, increased temperature within cities due to a dramatic increase in materials that absorb and retain heat, like buildings and pavement, on animal evolution. You have chosen a common species of European land snail as your study organism, since they are abundant and easy to spot, right? Because their shells are large and brightly colored. To perform your study, you enlist members of the public to take pictures of any of these snails that they see throughout the city and upload them to your database. You go through the pictures and count the number of snails with each shell color. The shell color in these snails is controlled by a single gene with two alleles, R and Y. These alleles show an inheritance pattern of incomplete dominance, so both phenotypes are blended in heterozygous individuals. So if you were to have a snail with the RR genotype, then you would have a snail with rose-colored shells, which is kind of pinkish in color. If you had a snail with the RY genotype, you would have a snail with a brown shell. And the YY genotype corresponds to having yellow shells. When collecting the data, or when your data collection period has ended, you are left with the following data as you see on the screen. We will use this data now to determine whether this population is at Hardy-Weinberg equilibrium or whether it is experiencing evolution.